So, welcome you all. Today, in this auspicious Republic Day, in the morning, we have actually remembered several heroes of our country. And now, today, we are actually going to commemorate the first death anniversary of our beloved Dr. Bhan, whom we lost last year in the same Republic Day. So, in this first death anniversary, we pay our homage in our own way to this great scientist, the inspiring leader, the icon of dynamism, and the architect of modern biotechnology. Whenever I think about Dr. Bhan, it always reminds me his affectionate touch and the inspiring words. Among many, he has built up many institutes, he has built up, mentored many scientists. I am one of those very, very fortunate individual whom he has ensured to grow, to contribute to the translational research. In fact, actually, I was working on protein synthesis, that is translation, and it is Dr. Bhan who made me to work on translational research. So there was a transition from translation to translational research in 2008 when he actually thought there is an urgent necessity to set up a center of excellence for hepatitis C virus research in the country. And today he would have been very happy to see that the research of hepatitis C virus is acknowledged with the Nobel Prize this year. I'm sure he is watching from the top. Not only that, that's his actual vision. Not only that, 12 years back, he thought that the dream that Professor Partho Majumdar is having to set up a center for biomedical genomics is going to be the future. And today we all realize and appreciate the need of this biomedical genomics for the era of precision health management, particularly when we are facing this worldwide pandemic situation and the way DBT has showing that how India can respond, and under the leadership of Dr. Renu Soru, DBT has actually shown today that what is our ability to deliver and actually stood up to the promise and the expectations of the country people. And we are very happy. I'm sure Professor Bhan would have been very delighted to see the tableau of this Department of Biotechnology in today's Republic Day parade. So we are all missing Dr. Bhan, and I'm sure all of us are blessed, and India could make a vaccine racing with the world with so many other stalwarts. India has also demonstrated their ability to come up with our own products of detection kits, vaccines. We have some of the lead antivirals, all are developed, and Department of Biotechnology has contributed heavily in this endeavor. So I'm sure these are all because of his blessings, and that he has inspired the pool of scientists and made sure they can lead from the front. So this is the best way we thought. Actually, it's actually the idea of Professor Partho Majumdar to pay tribute to this great scientist, the great visionary, and commemorate his wonderful science, his contribution in the science and developing science and the technology, in the particularly in the area of biotechnology. Thank you all for joining this event. I'm sure all who are actually watching this live event will be actually witness the befitting way of our homage to this departed soul, and we actually commemorate and inspire ourselves again for his dream, and we'll achieve those dreams collectively. And with this few words, I would like you to thank you again for joining this event and welcome you all. Now it's all over to Professor Partho Majumdar to continue this event, and he will moderate the sessions. Over to you, Partha.
Thank you so much. Thank you so much, Shomitra. It's really an auspicious day in some ways that one of our heroes, Dr. M.K. Bhan, is with us today, or we are going to pay our tribute to him today. Some people leave their footprints on the sands of time, and he is one of those people who has left his indelible footprints on the sands of science and clinical research. Let me welcome all of you who have joined this morning's event. In particular, let me welcome and express our gratitude to Mrs. Bhan, Shireen, Abhishek, and Gautam for having joined this event. We have conceived of this event. We had conceived of this event or thought that we will be able to get together physically, but that has actually not been possible for reasons that are clear to us. Be that as it may, it's a great honor for us to have some of the students and followers of Dr. Bhan who are actually continuing to leave the trail, his legacy, and move the torch of his legacy forward. We have a few speakers whom I will introduce as I move on. But I would like to again restate that Dr. Bhan, first of all, was one of those characters who was very loving, who was a profound thinker, and who actually liked to encourage people from the front. We have seen that over the years in our scientific advisory committee meetings of the National Institute of Biomedical Genomics. I have witnessed his caliber and how he encourages people from close quarters outside of this institute as well. I've had very close relations with him, even though I was not mentored by him, but I feel that I've been blessed in some ways to have been, in association with him, and I've learned a lot from him. This institute could not have been created without the tremendous capabilities and the support and everything that he provided to the formation establishment of this institute, right from the beginning, when we didn't even have a square inch of land, negotiating with the government of West Bengal, negotiating with the government of India, and helping to get faculty members and get other people into the institute such that institute can function. So this institute completely owes its existence to Dr. Bhan. And I personally am very, very grateful to Dr. Bhan, not because we have together established this institute, but more importantly because the kind of philosophy that he had, philosophy of life, has left an indelible mark on me. So it's sad for me to be able to sort of preside over this particular event. But I didn't imagine that I would have to do it. But this is the best way that I can pay, and we can pay our tribute and homage to a departed soul. But I think that he still lives with us, and on a daily basis he provides guidance to us. And it's really very nice to have his legacy continued and his philosophy. In those of us who have imbibed his philosophy continue to live with it and propagate that philosophy into those of us, into those who actually would like to hear about his philosophy from us and see the fruits of that philosophy blossom in institutions such as the National Institute of Biomedical Genomics. So let me first tell you that Dr. Renu Swarup was to give the formal lecture in this tribute meeting. Today is the Republic Day. The Department of Biotechnology has a tableau that's on the streets of in the Republic Day parade on the streets of Delhi, and the Secretary, Department of Biotechnology, Dr. Renu Swarup, has been. It's her obligation to be there, sitting on the fences of the Republic Day parade. So that's where she is, and when she informed me about her inability to be in person, she herself offered that she will create a video or record a video and send it to us on this occasion, which she has. And I would request Bikram to play that video for all of us. Bikram?
Okay.
2020, a year which all of us will remember. 2020, a year which all of us will remember as the year of the pandemic, a year full of disruptions, a year which put us through a lot of distress. There were economic challenges, there were travel challenges, there were health challenges. But for many of us, 2020 is a year that we lost a legendary scientist, a person who was dearly loved by the scientific community, the medical health community, the researchers, Dr. M.K. Bhan. We lost him on 26 January 2020, a year when India celebrates its Republic Day. And that, I think, was his tribute to the country that he loved. Dr. Bhan has played a pivotal role in the growth of the biotechnology sector, in the growth of the medical research sector within the country. His entire lifetime, he dedicated to science, to clinical research, to bringing biology and clinical research together, and to taking the fruits of science to society. I recall my association with Dr. Bhan when I was in the Department of Biotechnology. I joined in 89, but I think my first acquaintance with him began somewhere towards the early 90s when his association with DBT for the rotavirus vaccine was gaining momentum. At that point of time, my discussions with him or my interactions with him were minimal, since I was not really looking after the medical biotechnology area directly. And as one of the younger junior officers in the department, we did get an opportunity to interact with the senior experts who would come in. And also, since I was responsible for the biotechnology vision in 2000, the coordination, it did give me an opportunity to discuss with many experts of our scientific advisory committees, our task forces. But when Dr. Bhan took over as the secretary of this department in 2004, that gave me an opportunity to work very closely with him. It gave me an opportunity to understand his way of functioning, his thought process, and also work with him very closely and see how he grooms and mentors the younger lot, the leadership qualities that he had, and more importantly, the wonderful human being that he was. From 2004 to 2012, his tenure as the secretary of this department saw transformational changes in the biotechnology sector, in the department's activities, and I think that was a true exhibition of the visionary that he was. We brought out our first strategy in 2007, and from 2004 to 2007, Dr. Bhan had worked with all of us very closely, deeply engaged with all the stakeholders to see, as we are moving in this growth of the biotechnology, what are those missing gaps which need to be supported? And clearly, an area that he felt required a lot of attention was the area of translational research. And that's how the translational ecosystem within this country took shape. Engaging the startups, engaging the industry was again a major initiative that was taken. And that is how we came up with BIRAC, an organization that we see today making so much of a difference. The bioclusters that we set up across the country, and our autonomous institutes focusing on translational research. THSTI, the Animal Biotechnology Institute, and the NIBMG, many others, which took shape as a part of that strategy of taking research through translation to be able to deliver the fruits of science to society. BIRAC is something which is very close to my heart as well, and brings back huge memories of how I used to sit for hours and hours with Dr. Bhan and the group to be able to conceptualize this very new concept. I do recall that when we brought out the 2007 strategy, and since I had the good advantage of being the member secretary of that group, there was one clear indication we gave, and that is we must engage in public-private partnership. We did realize there was a vacuum in the whole startup ecosystem. And we said BIRAC would be set up as a separate vehicle to promote this. And then we really got thinking as to what shape BIRAC needs to take. Another wonderful quality of Dr. Bhan, being a creator, being a thinker, he gave a lot of freedom to the younger generation, to the younger colleagues, to be able to throw out their ideas, which he would then cherish, but fine-tune and give them back to you to see how you could implement them. I thoroughly enjoyed working with him on creating BIRAC. Today, when we see BIRAC standing as a strong foundation, as a strong pillar of supporting and creating this whole startup innovation ecosystem within the country, and the thousands of startups who have now come within the fold of BIRAC, who have been supported and mentored, and those large number of students who are on their way to becoming startups and entrepreneurs, it gives immense pleasure to sit back and think that this really is the foundation, this really is the organization that was envisaged when we began thinking about it in 2007. A great tribute to Dr. Bhan would be that this organization flourishes and this organization delivers beyond expectations. And I'm confident that the way it is structured, it will. The translational ecosystem of the Department of Biotechnology, the THSTI, one of his very, very pet institute, the NCR Biocluster, the Bangalore Biocluster, CCAMP, all these instruments, vehicles, and tools which helped promote the startup translational ecosystem. Today, in our S&T response to COVID, the key role which has been played by this translational ecosystem is definitely a tribute to Dr. Bhan. When we talk about India's response to the vaccine development for COVID-19, not a single day goes past when we do not think of him, when we do not fondly remember him, but more importantly, each step that we take, we feel that his guidance, his blessings are with us. I'm sure he would feel proud of these institutes who have today delivered, who have stood up to the challenge which has been posed before them. Had this ecosystem not been there, it would have taken us very long to come forward to respond. So it's the visionary thought which created this very strong foundation, which today could stand up and respond to this challenge in a very short period of time. Today, all the vaccine work that you see happening within the country, the industries are partnering with our institutes to be able to deliver. We are very pleased that THSTI has been recognized by CEPI as a global immunogenicity laboratory. And around this, we are now setting up more. The biorepository, excellent work which has been done around COVID. Of course, the earlier work which they've delivered for many other activities, again, is something which he was always very proud of. But I'm sure this would be another such achievement which would have made him proud. I can go on for hours and hours recalling those wonderful times that we have spent under the guidance and in association with Dr. Bhan, working on many, many documents, working on many new thoughts, putting final shape to just ideas which came up at that point of time, which today have enabled the growth of the biotechnology sector as we move forward. But I think I would just like to close by saying we do miss him each day. We remember him fondly, but as he would have always liked it, let us move on and carry on this journey with complete dedication and with this commitment that we are competent to be able to deliver. We have the level of confidence to be the best, and definitely we will be able to carry this confidence with us to be recognized amongst the global best. So I would just say thank you, Dr. Bhan, for leaving this legacy behind. And our commitment is that we will carry this forward. Thank you so much. And thank you, Dr. Partha. Thank you all the scientists for arranging this very important discussion, this meet of the scientific community as a tribute to him when we complete 1 year without his physical presence with us. Thank you so much.
Thank you very much, Renu, although she can't hear, but through all of you, let me convey our gratitude to Dr. Renu Swarup for this wonderful recapitulation, recollection of what Dr. Bhan has done and her own association and learning from Dr. Bhan. I know that there are many young people in the audience who may not know who Dr. Bhan is, so let me just quickly tell you that Dr. Bhan was born when India got her independence. He was born on the 9th of November, 1947. He passed away, as you have just heard, on the 26th of January, 2020. He did his clinical work, clinical training at the Armed Forces Medical College. And when I invited some of the members of the Tata Medical Center here in Calcutta, Dr. Mishra reminded me that Dr. Bhan was his senior in AFMC Pune, and they used to call him Maharaj Sir. Many people actually called him Maharaj, and indeed he was Maharaj of ideas, of love, affection, and guidance. Dr. Bhan then went on to spent most of his life at the All India Institute of Medical Sciences. He was a pediatrician. He was a clinical researcher. The areas of research clinical research where he left indelible marks. Today we will hear that from several people. But before we get into the science, there's also another area. Dr. Bhan actually touched upon many, many areas, but he was very keen on training. And one of the major impactful training programs that he had initiated jointly with the Wellcome Trust in the UK, which is called DBT Wellcome Trust India Alliance. The first CEO of the DBT Wellcome Trust India Alliance was Anuradha Lohia, who is currently the Vice Chancellor of Presidency University. She was not only the CEO of the Wellcome Trust, DBT Wellcome Trust India Alliance, but even before its formation, she was in intimate touch with Dr. Bhan, exchanging ideas as to how to move this forward, etc. So I requested her to describe that to us, and she happily agreed. And so it's my pleasure to introduce Dr. Anuradha Lohia and request her to give this, share this information with us. Anuradha.
Thank you, Parthuda. A very good morning to all of you. And I would like to pay my personal tribute to Dr. Bhan. And I thank Parthuda for giving me this opportunity and inviting me to speak at this auspicious occasion. Thank you, Shoumitru. Thank you, DBT, and all of you who are listening today. I'm indeed glad to be a part of this beautiful occasion and to share this moment with all of you. I never knew Dr. Bhan personally or was mentored by him scientifically, but of course we all knew Dr. Bhan because he was larger than life. And the first time I remember I met him many, many years ago in NICET, Kolkata, where he was talking about the rotavirus vaccine and it was ready to be taken to the Bharat Biotech and other agencies. And I was fascinated thinking, also asking, will this really work? Nothing actually works. We talk, but does it really go forward? But I was also very young and I had just started my own research career. So I think my cynicism, my trepidation, my apprehension were very well answered when all this became reality. I must tell you, I was very stunned when out of the blue, I, who had no formal connection or informal connection with Dr. Bhan, when I was asked to become the CEO of the India Alliance. And I really didn't know why or how was it me. But thankfully, because I was so stunned, I didn't ask too much. And when I met Dr. Bhan, he didn't even give me a chance to express any doubts because he was very clear in his head what he wanted, what was to be done. And in his very inimitable way, he started by telling me, because I did ask him, but how is it, how did it all start? And he said, well, I was at a meeting in London and I was talking to the director of the Wellcome Trust. And that's where they were discussing. And this idea apparently was verbalized by Dr. Bhan first time in that meeting when he told Mark Walpert, the then director of the Wellcome Trust, that we would like to have an alliance in India with you. Your fellowship for young scientists has served us well. We have got some excellent candidates who come back to India with the Wellcome Trust Fellowship, the competitive ones, and we would like to have a similar model with you as a partner in India. So I think there was some conversation about the money and Dr. Mark Walpert said, well, we can only spare this much and how much. And what I consider a landmark moment in my understanding and my knowledge of everything was the audacity and the courage with which Dr. Bhan told Dr. Mark Walpert, we don't need your money. We need your brand. We need your image. When he related it in his usual relaxed smile and, laughing at one side, I was so stunned. I think that memory, that image of him saying it to me will remain with me for life. And I just thought, really? Do we dare? We are poor, but clearly he was not. And we are not, because we went in for a 50-50 partnership. How audacious can we be? And today, several years down the road, I understand DBT is going to be 100% of the funds provider. This is Dr. Bhan. How audacious, how beautifully courageous. He certainly inspired me to go beyond my comfort zone in every direction. You can go beyond your comfort zone in a zone where you are knowledgeable or comfortable. But to go beyond your comfort zone where you know nothing, it's like leaping without looking. I think that was Dr. Bhan's inspiration. So when I started to feebly express my inability to do any administration, he pretended he didn't even hear me and he said, "You go and meet Dr. Mark Albert tomorrow." So I didn't know whether he was just ignoring me or what it was, but I realized that was his way of giving me confidence. And I felt extremely committed to make this happen. I must tell you all that at that time, all my colleagues, other well wishers, everyone said, "This won't work. This won't happen. I mean, you can start as many good things and it will flop." And I had this image of this man telling Dr. Mark Walpert, who was fairly, he had his own complacency about what the Wellcome Trust was, who was a giver of funds, to be told, "We don't need your money. We just need your brand." I think this will live with me forever. To have faith in myself, to have faith in us, to be strong. And the only way the strength comes is when you believe in it and you go ahead and do it. And this is what Dr. Bhan helped me to achieve. So I really, Dr. Bhan, today I bow my head to you and tell you how wonderful it was to get a new lease of life, a different plane where we all evolved mentally. And I'm so proud of all the Wellcome Trust DBT India Alliance Fellows who we've been able to bring back since 2010. Thank you, Dr. Bhan.
Thank you, Anuradha. Thank you very much for those kind words and recollections. We now have a series of science expositions. All of the people who actually have been very closely associated scientifically and personally with Dr. Bhan are going to give a series of talks. Dr. Bhan actually touched many areas of science, but he has left his imprint in certain areas more than in other areas. And we don't have the opportunity of touching all the areas where he made an impact, but his greatest impacts have been in certain areas. And we've chosen those areas to showcase. And not just showcase what Dr. Bhan did or what Dr. Bhan started, but more importantly, where are these domains marching forward and how are they marching forward? So we've called this exposition as March of the Domains. It's essentially to showcase where what Dr. Bhan started, how the legacy is marching forward. The first person who will talk to us is Dr. Neeta Bhandari. She is the torchbearer of a major legacy that Dr. Bhan started, which is Society for Applied Studies. And she's the torchbearer of the Society for Applied Studies. She is going to talk to us on interventions to reduce stunting in young children. What are the opportunities and challenges? And she has worked with Dr. Bhan for a very long time. She herself is a clinician, is a doctor, and worked at the All India Institute of Medical Sciences, did her PhD with Dr. Bhan, and has continued to work on various pediatric problems and problems of the youth. And she will describe to us what Dr. Bhan started and where that particular domain is going. Neeta, all yours.
Thank you. I will be talking about child undernutrition today with an emphasis on linear growth. This is not intended to be a review of the subject, but I only want to illustrate using one example how one can generate evidence in an area. The work that I will present today was all done under Dr. Bhan's guidance. So childhood stunting, we all know the consequences, whether short-term or long-term. Short-term consequences on health, increased mortality and morbidity. Longer-term, reduced adult stature, increase in obesity and associated comorbidities, impact on reproductive health. Similarly, in development, in the short term, reduced cognitive, motor, and language development. But as these children grow older, reduced school performance, learning capacity, and unachieved potential. Economic, increased health expenditure on ill children. But as they grow older, studies have shown that these adults have reduced work capacity and work productivity. So these are some projections that were made a few years ago, which show the declines, the prevalence and trends of stunting in preschool children. And if you see the data for Asia, you will see that there is a decline, but the decline is not as much as desired. National surveys in India, you will see the same situation again. There is a decline in the prevalence of stunting. The NFHS 5, I haven't shown the data because these are not complete. They are only available for 22 states. And even in those, in some of the states, it is found that stunting rates have increased. So when we review this, acceleration in reduction of childhood stunting is a widely accepted priority. And the question is it feasible? And if so, how? So these are lessons from some community trials that we did to address malnutrition in North India. The study started in the mid 1990s. And the first one we did was in an urban slum in South Delhi, opposite. There used to be a big slum opposite the Lotus Temple in Delhi. And we enrolled 418 infants aged 4 to 6 months old. And we had 4 groups. One group got food supplement, which was a packet prepared by the National Dairy Development Board. It had 220 kilocalories and had added micronutrients. Along with that, mothers were counseled on how to give the packet. The second group was counseling alone. The third was a visitation group because we know that there may be an unintended beneficial impact of visitation. And the fourth was a no intervention group. And weights and lengths of these children were taken every 3 months. So what we found is that reported energy intakes, and this means we had asked mothers what children had consumed and collected empty wrappers, but the reported energy intakes increased maximally in the first group, which was the food supplement and counseling group. They also increased in the nutrition counseling group, the second one, compared to either of the 2 control groups. We found that in the first group, breastfeeding rates reduced. There was an increase in morbidity. In terms of weight gain, there was a 250 gram additional weight gain in the food supplementation and counseling group, but no impact in weight gain of nutritional counseling alone. There was a 0.4 centimeter length gain between 4 and 12 months in the food supplementation and counseling group, but this was not statistically significant and smaller than desired. Then we did a behavior change intervention trial in rural areas of Haryana. This was a cluster randomized trial. We developed feeding recommendations locally based on the foods that were available and acceptable to families. This was done through government workers, Ashas, Anganwadi workers, ANMs. The workers were trained, and we enrolled, the research team enrolled and followed up a cohort of newborns, around 550 in the intervention clusters and 550 in the controls. So this is just to show you the opportunities or contacts at which we counseled. Children were counseled at primary health centers when they were taken there by their families for illness. They at Anganwadi centers when they were weighed, at immunization sessions, at community meetings. Haryana used to have a monthly community meeting. The messages on optimal complementary feeding practices were conveyed during those meetings, during monthly home visits of Anganwadi workers through traditional birth attendance. This study actually aimed to improve exclusive breastfeeding, so breastfeeding and complementary feeding practices, and that's why you have the DAI also involved. What I'm presenting now is data from 6 months onwards. Even the private practitioners, the local private practitioners were involved so that they could give these messages at sick child contacts. So what did we find? Overall, there was no impact on weight gain. We had higher attained length at 12 months of age and higher increments in length between 6 and 12 months of age, and I've highlighted these two. On side by side, we felt maybe it was individual micronutrients that had an impact on growth. So beginning from mid-90s, some studies were done looking at the impact of individual micronutrients on growth, impact of vitamin A on physical growth of children. All these were randomized controlled trials, all done in populations. For vitamin A, overall, no impact in increments in weight and lengths. The zinc supplementation, this was perhaps the largest trial that examined the impact on growth to around 2,500 children given zinc or placebo daily for 4 months. There was no impact on weight or length gain. We also did a trial on low birth weights enrolled from the hospital, but followed up at home for till 1 year of age. And these were 900 low birth weights given daily supplementation, zinc or placebo, till they were aged 12 months. But what we found was weights and lengths were similar at 3, 6, 9, and 12 months. So why was the impact less than expected? Was it that the intakes were overreported by mothers? Because when you counsel so much, mothers, know what their answer should be. Did the diets, whenever we gave a supplement, did they displace the routine diet that the child would have got otherwise? Was it an issue that families were not giving animal origin foods? Because in Haryana, what we had found, these were primarily cereal-based diets. Children were given very small amounts of milk. Was the issue that there were additional energy and nutrient requirements in the presence of infection and stress? Were there other linear growth relevant nutrients that we were missing and were not in our supplements? And some studies that have been done since then have shown that stunted children have lower levels of choline, lysine, and essential amino acids. Was clinical or subclinical infection responsible? Was it the gut microbiome? Or is it that non-nutrition interventions like water supply, sanitation, care and support, are these important? Another issue was that did we need to start these interventions earlier than before birth? So was intrauterine development inadequate because of maternal malnutrition? Should we intervene before birth? Or do we even need to intervene before conception? because pregnancies are reported late. So if you intervene during pregnancy, you may still not end up delivering all the interventions that you need to during pregnancy. So a few years ago, all of us met and reflected on what now. This was about 4 or 5 years ago. We all recognized that poverty alleviation and equity, these were paramount. Equity in, all the programs reaching the most vulnerable. But meanwhile, could we identify and test a package of the most critical, high impact interventions and deliver it in a way that if it were successful, it could be delivered through programs, and the package would be intended to reduce both low birth weight and stunting at 2 years. So our hypothesis was within a single generation, substantial reduction of stunting rates is feasible if major factors for poor linear growth are addressed concurrently, which is health, nutrition, environment, psychosocial care. If the interventions are initiated during the preconception period and continued throughout pregnancy and the first 2 years of life, and the delivery of the interventions is such that it enables high coverage with quality and equity. So the study design principles were we should be able to measure the independent effect of preconception interventions, of delivering interventions in the preconception period. We should be able to measure the combined effect of interventions being delivered across preconception, pregnancy, and the postnatal period. If we saw an impact, we should be able to examine the effect stratified by maternal height to understand intergenerational effects. And our delivery mechanisms should be optimized in a way that we could link home and health facilities. So this is an ongoing study. It's Women and Infants Integrated Interventions for Growth. It's called WINGS. And what we are starting in preconception, and children will be followed up till 2 years of age. I've mentioned the primary and secondary outcomes. These have all been published. The methods have been published. The study will finish in probably by the end of 2022. So what this complicated design shows is what I want to demonstrate here is that we identify eligible women through a door-to-door survey in urban low resource areas, and then they undergo, those who are willing undergo a first randomization stratified by maternal height, and we have enrolled 13,500 women. These women get either the preconception intervention package or routine care. And once they become pregnant, they are randomized again. Now, those who don't become pregnant are followed up for a maximum of 18 months. So once they report pregnancy, they are randomized to get either the pregnancy and early childhood interventions or routine care. And this is then in both these groups, the routine care group initially, and the one that got routine care initially, and the one that got the pre and peri-conception package. So because of these two randomizations and the factorial design, at the end of the study, we will have 4 groups, 4 analyzable groups. Group A would have got interventions throughout. Group B only in the preconception period, Group C in pregnancy and post early childhood period, and Group D, no interventions throughout. So the study is in low to mid socioeconomic neighborhoods, and the reason, the main criteria for choosing this area was that the stunting rates were closer to the national average. So how did we select the interventions? We did a literature review to examine interventions for reproductive age group women, mothers and children, and the possible effect on length at different ages, birth weight, size for gestational age. We chose those where the magnitude of impact was significant and the quality of evidence was high or moderate, and we had a technical advisory group to guide us. So I won't go through the details. These are the methods are published, but the interventions are in the pre-pregnancy period, pregnancy, early childhood, and for mothers in the postnatal period, and in the domains of health, nutrition, psychosocial care, and WASH. There are also sub-studies embedded in the wings. So we are examining the effect of interventions delivered throughout the continuum, which is Group A, on breast milk quality, systemic inflammation markers, gut microbiome, and insulin-like growth factor at 6 months of age compared to routine care. And we are also doing body composition at 1 month of age in all the 4 groups. So what do we hope to learn from the study? This study, along with many others that are ongoing in different parts of the world, is expected to improve our understanding of this area substantially. We will know whether the impact of a preconception intervention package, what impact does it have on health and nutritional status of women when they get pregnant, on birth outcomes, and on length at 24 months. We will also know the impact of the package if delivered during pregnancy and early childhood period, or if it is delivered throughout the continuum. What would the impact be on birth outcomes and attained length at 24 months? If we do see an impact, we'll be able to examine it stratified by maternal height and by gestation and size at birth. All these women have ultrasounds, so we have a reliable estimate of preterms and small for gestational age. So as the trial is ongoing, we are learning some lessons, and learning lessons as we go along was something that Dr. Bhan was very keen that we do. We had intensive reviews every 2 to 3 months, even if it meant that, the investigators, 6, 7 of us had to fly to Geneva to discuss this with Rajiv over a weekend because Rajiv was not available to fly to India at that time. But till last year, we have been doing this every 2 to 3 months. So currently interventions are delivered in the 1,000-day window, pregnancy and early childhood. And is the window sufficient? It does seem that it is not, because what we find is that 3/4 of women have moderate or severe morbidity during the preconception period, and 2/3 of them respond within 3 months of management. If you're interested, this paper has been published. Can we deliver antenatal care better? We have a collaboration. We have investigators from Safdarjung Hospital, and when the study started, all women who became pregnant were being referred to Safdarjung Hospital for all antenatal visits. And we were not getting very high compliance rates because women couldn't be away from home for 4 to 6 hours. So we set up an outreach clinic in the communities And what happens now is that those who are non-high risk, or those who want their supplies to be replenished, the supplements to be replenished, or for want to come for follow-up of, let's say, thyroid disorders, reproductive tract infections, they come to the outreach clinic. And this has changed everything. Our compliance rates for these are now 75 to 80%. Our median number of antenatal visits is 8. So we've also gained a lot of understanding around gestational weight gain, and because we are taking monthly weights during pregnancy, we found that wasting and stunting rates are high in less than 6 months. And this resulted in us submitting a concept note for another study to see how we could do better in this area. And we've also learned a lot on managing inadequate weight gain between 4 to 6 months in exclusively breastfed infants. So what I have described till now is an illustration to show you the process that one can use to generate evidence in one area. There are several such examples on zinc, ORS, rotavirus vaccine, and several others. As I was working on this presentation, a few messages came to my mind, and some of these were the first for researchers, that research should lead to solutions that can be applied, and the inverted commas show these are actually what Dr. Bhan used to say. We need to define the right question to answer. He always felt that you can't think in isolation or alone. Ideas can't come from one. We need to collectively discuss issues with the best mind. The target he set for all of us was to aim for mastery in thinking and in doing. And most important of all, read what others are doing, synthesize and connect. The connect part, I think many of us don't do this too well, but he was brilliant at linking whatever we read, whatever he read, with other work or our work that was going on. Nurturing teams and young folks, as he called them, was very important. I won't talk about this because we have two people talking. Love to have fun, and Shinjini will identify with this many times at her expense. So most of the time it was at other people's expense, but I think in particular he loved pulling Shinjini's leg. Collaborate, not compete. Here I will tell you a very short story about conflict resolution. We were all at AIMS together, and as is usual in teams, there is often conflict between peers. And the background is that he had a small room in AIMS, no place to sit. We only had those high stools, the lab stools where your feet don't touch the floor. At the end of the day, if he had sensed some conflict, we would be called to his room. We would sit in a circle. He would order tea and vada samosas. I don't think we ended up discussing the conflict that much, but at the end, he would ask us to hold hands. And this is like you do in, Ringa Ringa Roses, everybody sitting in a circle. It felt very foolish then, but I think something worked. We are all now in different institutions far away, but the deep caring and commitment we have for each other is rare. And above all, what he felt was that science is for human welfare, and you can't do this work unless you care deeply for those who we claim to serve. So I will end by his quote. His aspiration for India's children. And this he said in while he was addressing a group of young students. So what he wanted is that every child born in this country, when in the mother's womb and until they are 18 years of age, their brains are well developed, their body is well formed, they get good education and nurturing care, and they become creative, enjoyable, and responsible citizens. Thank you.
Thank you very much, Nita. That was just absolutely wonderful. Yeah, he had his charm and he had his own ways of resolving conflicts. Very, very nice. We will not have any questions in this entire session, primarily because we will then not be able to keep to time. So those of you who have questions may actually send by email to the respective speakers. Our next speaker is Professor Gagandeep Kang. Professor Gagandeep Kang has been associated with Professor Bhan for a very long time, and she represents an area where Dr. Bhan had actually made a major impact, both in terms of innovation as also in terms of saving lives. And this is the rotavirus vaccine. So I will request Gagandeep to tell us about vaccinology with special reference to the development of the rotavirus vaccine. Gagandeep.
Thank you, Parthuda. Can you see my slides?
Yes.
Yes, we can see, ma'am.
Okay, right. Thank you. So I'm going to be telling you about vaccines in the context of the rotavirus vaccine story. And that is one that is indelibly associated with the Dr. Bhan. So this is an outline of my talk about India's place in the world of vaccines, then really about rotavirus and a summary at the end. So if we look at vaccine research overall, if you don't look at India and you divide up the industrialized world and the less developed world, in developing countries, we have the bulk of the population, we have the bulk of the disease burden, but we actually have a smaller proportion of vaccine usage in our populations. If we look at India, we are somewhat unusual in that we make the most vaccines in the world. About 60% of the vaccines procured by the Gavi Alliance, which provides vaccines to low and middle income countries for their routine childhood immunization programs, are made in India. This is about 40% of the global vaccine manufacture by doses. It only comes to about 2 to 3% of the global valuation of vaccines. So we make a high volume, but until now, we have largely focused on low value products. But with SARS-CoV-2, the landscape is changing. The world is looking towards India as being a major supplier of vaccines. And I think many of the things that we are doing today, many of the companies that are at the forefront of developing new vaccines would not have been where they are without Dr. Bhan. Both before he went to DBT and after DBT, he played a major role in supporting the industry. I don't think there is a single company or a single leader in the vaccine space in India that has not been affected by Dr. Bhan, where he has not played an immensely supportive role, even in companies that don't yet make vaccines such as Minvax with Raghavan Vardarajan, seeking to develop new vaccine candidates for influenza and for SARS-CoV-2. But within DBT, also, Dr. Bhan played a major role. Much of the work that he did was supported by the Indo-US Vaccine Action Plan in the early years, and then he amplified the impact of the Indo-US VAP well beyond rotavirus vaccines. There have been many other missions that were launched at DBT during his time, and he played a key role in the development of BIRAC, which is home to the National Biopharma Mission and to INSEPI. He was on CEPI's Scientific Advisory Committee and played a large role in having it accepted within the Indian ecosystem. So if we come back to rotaviruses, rotavirus is something that I think was a passion for Dr. Bhan. From the very early days, the virus itself was described in 1973, and in the early 1980s, he began to study it and then continued to study it throughout his career. If we look at rotavirus before the vaccine started to be widely used across the world, you can see why studying rotavirus in India was critically important. Globally, about 40% of all diarrheal deaths are still attributed to rotavirus. And if you look in India and we look at severe disease, the ICMR National Rotavirus Surveillance actually came from a program that Dr. Bhan started to champion in 2001, persuaded Dr. Ganguly that this was an important endeavor to undertake. And this surveillance ran for the next 15 years and showed that until we introduced vaccines, 36 to 40% of all children hospitalized with diarrhea across the country had rotavirus. Now, given our disease burden, obviously it was clear that we needed a vaccine, but which vaccine would it work? Would we be able to afford it? In the late 1990s, Dr. Bhan was planning to evaluate a vaccine called RotaShield. And that was a vaccine that had been developed by Wyeth. And the idea was that this was a vaccine that might be suitable for use in our country as well. So they were getting ready to do vaccine trials when there were some findings from the Wyeth study that resulted in the vaccine being withdrawn. This was a vaccine that was withdrawn because of intussusception. It was a vaccine that had been developed on an approach that had been championed for rotavirus vaccines, which was the idea that vaccines needed to have multiple genotypes because rotavirus surveillance had shown that there were multiple genotypes that circulated in many parts of the world. And in countries like India, the diversity of genotypes was greater than in other parts of the world. Developing countries had many types of rotavirus. Developed countries had few types of rotavirus. And based on animal studies, people thought that we needed to have vaccines that represented a wide array of genotypes. So RotaShield was withdrawn, and it was shown that there was a risk associated with RotaShield, which was an association with intussusception, particularly in the first week after the first dose of RotaShield vaccination. This led to the withdrawal of the vaccine, and Dr. Bhan repurposed the study of RotaShield, working with Neeta and with Rajiv to show the risk of intussusception in India, which was the first and paper that established a baseline for intussusception in India and led to further studies that looked at vaccine safety. So when Rotashield was withdrawn, there were questions being asked around the world. Would we ever have another rotavirus vaccine? And actually, there were studies that wound up being designed that were very large studies that led to the licensure of two vaccines made by GSK and by Merck. They were designed to assess a risk of intussusception that was similar to Rotashield. The studies were successful and they resulted in the licensure of two vaccines in 2006, but these vaccines cost about $200 per course. So these were clearly not going to be vaccines that were going to be suitable for use in India. And Dr. Bhan had by this time turned his efforts to developing a vaccine candidate on which he had been working from the early 1980s. In 1985, as a pediatrician at AIIMS, he had observed an outbreak of asymptomatic rotavirus infections in the newborn unit and found that there was a very high rate of infections among neonates that were hospitalized for a longer period of time. These were asymptomatic infections, and the strain based on electrophoresis, which was our first technique for being able to look at the relatedness of rotaviruses, which involved extracting RNA and running it on a gel and staining it with silver, it seemed that all the strains in this neonatal nursery were very, very similar. He then followed up these children who had had this infection with the strain that was identified to be G9P11 and showed that children with neonatal infections had fewer attacks of rotavirus diarrhea than children who had not been infected. And this led to the idea that children with neonatal infections were protected and therefore the strain could be thought about as a vaccine candidate. A little later, another strain was found in Bangalore that had a similar pattern. And this strain was also found in neonates, also seemed to protect from infection. But unlike strain 116E, which had been identified in AIIMS and studied very deeply under the Indo-US VAP and shown to be a human strain which had acquired a bovine gene, it looked like strain I321, identified at the Indian Institute of Science, was actually a bovine strain that had acquired some human genes. So I321 follow-ups, like the Delhi follow-ups, seem to show that this strain was also protected. So speaking to what Neeta said, instead of just promoting the strain that he had found, 116E, Dr. Bhan suggested that both strains be taken forward together as potential vaccine candidates under the Indo-US Vaccine Action Plan. Now, these strains were characterized in the US and in India, and it was hypothesized that both of these strains would do well because their mothers did not have antibodies against P11, which is the bovine protein that is found in both strains. So there was a lot of testing that happened in the US. There were pilot lots that were made for as vaccine candidates and for challenge studies. The strains were adapted to culture and they were characterized very, very carefully. Phase 1 studies were done in US adults, and there already there were beginning to be signals that 116E might be performing differently from I321. This was also seen in trials that were done in US children, and it seemed like 116E was producing a better immune response than I321. At this stage, the strains were brought back to India and were given to Bharat Biotech, and that's a dramatic story in itself. And Neeta and her colleagues at AIIMS then began to do the trials in India. Very briefly, phase 1, phase 2A studies, phase 2B studies were essentially done. Phase 2A with both strains, and then taking forward only 116E because the immune response to I321 was not particularly good. A dose was decided on for 116E, and then plans were made for evaluation of the clinical efficacy of this strain. This was the stage at which Neeta and Dr. Bhan came to Vellore and asked if we would also participate with them in this study. It was a matter of pride for many of us to be asked to work with Neeta and Dr. Bhan, and we were really glad to join the venture with the Society for Applied Studies and KEM Pune, where the phase 3 study was planned as a clinical efficacy study in 6,800 participants. There was obviously a lot of preparation, a lot of discussion, and because this was going to be a placebo-controlled trial, there was a lot of issues about what we needed to do in order to be able to look after the children whom we would recruit into the study and follow up for 2 years. This study was probably one of the most careful phase 3 studies ever done, and not just in India, I think for many parts of the world as well. This study ensured that children were looked after for the entire 2-year period, and that it really didn't matter what kinds of problems children had. It was the study's responsibility to ensure that the children were managed as carefully as possible. So I think a lot of us had doubts about whether rotavirus vaccines would work or not. And this was because we had learned from the experience with polio that oral vaccines don't have the level of efficacy that is seen in Western countries. And by the time we got our trial readouts for the phase 3, showing 55% efficacy, there were data that were available from Africa and from other parts of Asia with the multinational vaccines also showing low efficacy. There were lots of questions. There had never been a neonatal rotavirus vaccine candidate before. Given that it produced asymptomatic infections in neonates, would it work to protect children? We didn't have a correlate of protection. We measured IgA, but we know that that is not a correlate of protection. So the only readout that was reliable for rotavirus vaccine was clinical efficacy. And when the data were unblinded by the DSMB, we found that the vaccine did protect against severe rotavirus gastroenteritis and against hospitalized rotavirus gastroenteritis. And as far as safety was concerned, even though the study was very small, there was no intussusception that was seen during the 1, 2, or 4-week windows following any dose of vaccine. The unblinding happened for the interim analysis in 2013. The vaccine was licensed by the Drug Controller General of India in 2014. And not that Dr. Bhan's work stopped then. I remember being at the DCGI meeting By that stage, he was already in discussions with the Serum Institute of India on the second Indian rotavirus vaccine. And there were a lot of questions about what kind of support Serum needed for these vaccines. Serum was threatening at one stage to take the vaccine trial overseas if they couldn't do a placebo-controlled trial in India. Despite having developed his own candidate as a rotavirus vaccine, Dr. Bhan provided every support that it was possible to provide to Serum Institute of India to make the second Indian rotavirus vaccine possible. This was a strain that had been brought in from the National Institutes of Health. It was a strain with a very different formulation from the neonatal strain. He helped them design the studies, he helped them do the studies, and this was the second vaccine that was shown to work in 2017. Both of these vaccines were WHO pre-qualified in 2018. They are now available for the developing world. A couple of weeks ago, there was a paper that demonstrated that Palestine switched from a multinational country product to the Rotavac vaccine, and that has resulted in a cost saving for the country. Rotacil, Rotavac are both now being considered by multiple countries in Africa and are being rolled out to go beyond Indian shores. But within India also, there were other vaccine candidates that had a series of discussions with Dr. Bhan about being taken forward as additional products for the Indian market. I won't walk you through the details for Shanta, but actually Shanta was the reason that I first got to talk to Dr. Bhan about rotavirus vaccines. This was a candidate that looked very, very promising. It was similar to the Serum product. This was a vaccine that did well in phase 1 and phase 2 and then failed in an extended immunogenicity study, and the vaccine program was halted. There was yet another company in India that looked at making rotavirus vaccines. This was Hilleman, which is a joint venture between Wellcome Trust and Merck. And the idea was that Merck would provide Hilleman with bulk vaccine, and Hilleman would develop it as a heat-stable product. This also went through phase 1 trials, and the program halted in 2018. I highlight these to show that, sometimes it's very, very easy to talk about successes, but nobody actually talks about the failures. And if there was one thing that I learned from Dr. Bhan, it was that having multiple shots on goal is one way of maximizing the likelihood of success. So Rotavac and Rotacil became available, but the question really was, would this be taken up by the country? And when you consider immunization programs, the National Technical Advisory Group on Immunizations determines whether a vaccine should be taken forward. The questions are, is there burden? Is the disease a priority? Is it recommended by WHO? And would the vaccine promote equity? By this time, the vaccines were available in the private markets. They were not being used by public immunization systems. The vaccines were recommended by WHO, and we had done the work to show that there was a significant disease burden. Because there were ongoing safety issues, Dr. Bhan helmed a study with SAS, KEM, and CMC to do an early rollout, which was planned, and SAS and KEM were able to start that. CMC was a bit of a laggard, but we got there ultimately after the national program had started. And in March and April of 2016, India introduced the Rotavac vaccine, initially for 9% of the birth cohort, added more states, until by the end of 2019, Rotavac and Rotacil were being used across the country. We had at that time initiated studies to look at rotavirus vaccine impact assessment. How much disease does the vaccine remove in the real world? Is there a risk of intussusception? And if yes, how much of a risk? So we've been doing these studies based on the ICMR program that Dr. Bhan had helped initiate. And we were able to follow up 5 sites from the ICMR program in the pre-vaccine and the post-vaccine period. In the dotted line, you see vaccine coverage, and you can see what is happening to the annual peaks of rotavirus that we know occur in the winter. Overall, there has been a substantial reduction in rotavirus disease burden. We've also looked at intussusception, both in the early rollout study and in a separate study that we did as part of the impact assessment, and we found that there was no risk of intussusception with the Rotavac vaccine. We're currently doing similar studies with the Rotacil vaccine. So if I was to say what the rotavirus journey has been with Dr. Bhan, I think it was a promise realized after decades and decades of effort. But even more than what the vaccine has done for India and what the use of these vaccines will do in terms of alleviation of disease around the world, I think it's important to recognize that this journey built many, many, many people. It built partnerships, it built institutions, and that's a payoff that we are going to see, not just today, not just tomorrow, but for decades to come. So to end, I think there's never been anyone who did so much for India's children. It was a privilege to know and work with Dr. Bhan.
Thank you so much, Gagandeep. That was just absolutely wonderful journey. And as you correctly pointed out, that this is one man who actually impacted on lives of millions of children. And I'm so glad to hear that you've been a part of it almost from the beginning. Our next speaker is Dr. Shinjini Bhatnagar. Shinjini actually did her postgraduate medical degree with Dr. Bhan and then went on to do a PhD with him and has been associated with him for decades. Shinjini herself is a clinician, was at the Alvin Institute of Medical Sciences, and subsequently had joined the Translational Health Science and Technology Institute, where Gagandeep was the then director of the institute. So without further ado, let me invite Shinjini to talk about interdisciplinary platforms for deconvoluting complex phenotypes of public health importance. Shinjini.
Can you see my screen?
Yes, do full screen. Yes, great. Wonderful.
Thank you, Partha. Good afternoon, everyone. I'm going to talk about interdisciplinary platforms for deconvoluting complex phenotypes of public health importance. Complex title, complex domain. And I think I know why Partha asked me to speak about this is because it's important to share with the younger investigators how this complex program was conceived and actually made doable and simple because of Dr. Bhan. So this was pretty much my thought that I don't understand what this means. And when Dr. Bhan brought this discussion up with us, I said, I'm a pediatrician, I'm a pediatric gastroenterologist. What do I understand about complex phenotypes? And his response was that many of us are familiar with, many of us who have lived our lives with him for last over 3 decades. Be bold, explore things that scare you. Keep learning as much as you can about them until they no longer scare you. And for him, it was talk through these challenges so that you can turn your fear into excitement. And actually, that's what started happening. He always said that you have to be curious, you have to step out of your comfort zone, you have to ask thought-provoking questions. And these couple of slides I have actually stolen from one of his presentations. His discussions with us were, are we asking the right research questions? And are the right people asking the right questions? Are we just kind of rehashing what has already been done? Or are we stepping out of our comfort zones and identifying domains that need more knowledge? There are many unresolved questions there. And these have to have public health significance. Like Neeta said, that science has to be for people, to help people. There has to be scope for diversification. And that is the philosophy behind, as you just heard, Neeta and Gagandeep, on how these large hospital and population-based intervention studies were done. And this was his philosophy behind what we started as observational cohorts to study complex diseases. And that is the reason why he got all of us thinking. He said, look at preterm birth. We started to think. And I remember when Partha, who's another PI on this program, and Dinkar Salunke, another co-PI on this program, when overnight we had to sit together and discuss why this program has to be initiated. And gradually the discussions grew. For each one of us, it was getting out of our comfort zone, getting into areas that we didn't understand. But despite being Secretary, Department of Biotechnology, he was always there to take us through it. And I'm going to go through this program with you. It's complex, but learning from what Dr. Bhan taught us, that tell a story simply. Tell it the way you understand it. And so that's what my next 15 minutes are going to be. Preterm birth, as you can all see here, it was always believed that probably it's a disease of the West. And gradually one realized that in India, it is a problem. Large number of preterm births, many die soon after birth. A large proportion of total neonatal deaths are caused by preterm birth complications, and they have long-term consequences. When we started to do these brainstorming, and we realized that the effective solutions for preterm birth are not available because there are many unresolved questions, and this is largely because it's a complex syndrome of multiple etiologies. Genetic, biological, psychosocial, and environmental factors go into making up that preterm birth phenotype. And there are large fundamental gaps in understanding that. And that is because the studies that have been done have compromised sample size. There's phenotypic heterogeneity. Whatever clinical and biological assessments that have been done are like snapshot evaluations. The available evidence for mechanistic pathways has come from small animal studies. And really, there is much more to put together. And could the answer be, if this disease, which is a public health, which has public health significance, could it be, if it's contributed if we understand it in many ways, the environment, the clinical characteristics, the biology behind it, and possibly the omics technology to help us understand it better. And with all this, he was able to convince many of us that this is something that needs to be taken forward. And we started this program called Garbani. And again, for people who knew Sir, he never took names very well. So Garbani was never understood by him, but actually it was the Interdisciplinary Group for Advanced Research on Birth Outcomes, DBT India initiative. And he said, why can't you just call it this? Why this Garbani? Garbani today is a program which has gone on for about 5 years. We began with an interdisciplinary group which is comprised of, and again, unique in its collaboration, the research institutes, the DBT Autonomous Research Institutes got together, coordinated by the Translational Health Science Technology Institute, Regional Center for Biotechnology, National Institute of Biomedical Genomics, got together and collaborated with district and tertiary hospitals to take this program forward. Since since these autonomous institutes don't have a hospital of its own, we had to reach out to the district hospital, which had never heard of any research programs. But once again, we set up processes there, set up ethics committees there, set up labs in these district hospitals to be able to do this program. It is driven by a hypothesis that time series data on diverse variables consisting of clinical, environmental, genomic, epigenomic, metagenomic, and proteomic across pregnancy will stratify women into defined risk groups for preterm birth. Be able to do an effective risk stratification with modifiable and non-modifiable clinical factors, use predictive omics and image biomarkers to be able to predict women who may deliver early, and at the end, have an integrated decision algorithm for early prediction and timing of intervention for preterm birth. Once again, true to Dr. Bhan's style, it was not just a funding agency supporting it. It was a funding agency which put into place a steering committee, which was guided by Dr. Bhan to take this program forward. So at every step, he was there to help us understand each complex step and bring it to a situation where we could take it further. Another important thing that this program proposed to put together was set up a repository for biospecimens that were collected in this program, a large data management hub, clinical research units in district and tertiary hospitals, and above all, a young investigator program. So this was the concept of this program, and I will try to take it forward, explaining in as simply as I can in the next 10 minutes. So the deliverables were an effective risk stratification that facilitates timely referral for preterm birth, which would be a critical health system intervention for low middle income countries. And that is what we are striving to have algorithms for decision making from individual and population level risk stratification using interdisciplinary clinical, imaging, and biological sciences. A complicated deliverable. Use tools for more personalized and futuristic prediction of pregnancy outcomes with ultrasound using machine learning approaches. Develop biomarkers from simple biochemical markers going all the way to omics-based ones to provide potentially more customized decision-making tools for clinicians. And develop and augment research systems that can become national and global resources. We established a cohort of pregnant women in the district hospital. These women are enrolled early in pregnancy, not later than 20 weeks period of gestation, followed at regular time points across pregnancy, as you can see here, up to delivery and for one visit in the postpartum period. Detailed phenotype data is collected, as in socio-demographic, environmental, obstetric medical complications, drug and immunization history, and other parent and neonatal anthropometry. Biospecimens from the mother in terms of sera, plasma, saliva, urine, high vaginal swabs, neonatal heel prick, cord blood, and placenta, and paternal saliva is collected at defined periods across pregnancy. We do serial ultrasounds to document genetic chromosomal markers, fetal morphology and biometry, fetal-placental hemodynamics, placental position and morphology, and cervical texture and length. And this is done across pregnancy in the district hospital setting. The sample size initially was based on both cohort and nested case control analysis for documenting the risk predictors. And based on that, as of today, we have enrolled more than 8,000 women and documented almost 7,000 outcomes. And systematically, for the first time in the country, we have actually shown that preterm birth is a problem to contend with, 13%. And also the other adverse, it's also given us a platform to study the other important birth outcomes, as in stillbirth, low birth weight, and small for gestational age. So having documented that, knowing our conventional epidemiology and looking at simple analysis, we did risk analysis to see what would help in predicting these women early in pregnancy. And we found that many of the conventional predictors, as in prior preterm birth, short pregnancy interval, multiple gestation, short cervix, antenatal vaginal bleeding, were important predictors. But we also came up with some interesting risk predictors, like use of biomass, documented use of biomass fuel, exposure to passive smoking, and of course, some nutritional factors. This was a conventional epidemiological assessment. Now, when we did our analysis, we realized that these risk factors accounted for only 10% of preterm birth. And therefore, there was a need to identify more factors for better prediction. And that is where our whole philosophy and our hypothesis that to be able to improve the risk prediction, other predictors need to be studied, like image markers and the omics biomarkers. And that is where the whole interdisciplinary platform came into existence. And here, as you can see, it's a busy slide, but I'll just go through it slowly. We had our colleagues, our collaborators working in Regional Center for Biotechnology, looking at the proteomic biomarkers for preterm birth. And they first established that looking at what are the possible proteomic changes across normal pregnancy, and they have seen that there are salivary proteins and high vaginal proteins which are involved with certain biological processes across pregnancy, which can be identified as potential biomarkers and can explain some mechanistic hypotheses. Now they're working on identifying discriminatory plasma proteins between term and preterm. our colleagues in National Institute of Biomedical Genomics, using this platform of very well phenotyped data, have identified across pregnancy the microbiota of reproductive age is mostly dominated by Lactobacillus iners and crispatus, and are also excited to report that Lactobacillus iners is significantly higher in all trimesters in preterm birth, with Gaceri being higher in that in third trimester of pregnancy. So that what we are seeing as preliminary evidence that there are differences in the lactobacilli or in the microbiota in the vagina of women who are delivering preterm as compared to term deliveries. At National Biomedical, at NIBMG, our colleagues have found that there are antenatal epigenomic alterations in preterm birth, and specific biological pathways have been identified. And now the GWAS is being conducted using GSA with over 600,000 markers in 4,000 mothers, where genetic variants are being tested for association with quantitative outcomes like gestational age at delivery, birth weight, placental weight, and blood flow. So these analyses and this work is going on, but this would not have been possible if we had not set up an interdisciplinary platform where different disciplines using a common platform of well phenotyped data could be able to answer these questions. As we move on, the results from these individual omic platforms will be validated in another set of independent samples and population cohorts, and then there would be an integrated analysis to take it further. It's also interesting to note here that people like me who didn't understand what omics was when we started this work, I didn't know how it would help in understanding a disease, can actually sit here and try to present to you some of this work. And similarly, it's been really interesting to see Partha and his group, Dinkar and his teams, all of us sitting together and talking about, and they asking us questions on how pregnancy moves forward, what are the problems of labor, and how can these problems be solved, trying to answer some of these questions through biology and the omics technology. The next thing was, could we have use the ultrasound imaging for personalized tools for clinical decision? And here the applications of artificial intelligence is being used, and in collaboration, and again here, Dr. Bhan pushed us to go and talk to University of Oxford, the Department of Biomedical Engineering, and to understand how we could use their technology and their expertise for our data on ultrasound images to take it further for application for improving pregnancy outcomes. And today, using these AI technology, we have some evidence on looking at developing prediction models for preterm birth and gestational age, and also softwares which can automate measurements to reduce time scan done by almost 50%, which will help our sonologists at peripheral centers and reduce inter-operator variability. Of course, these models have to be validated further before they can be put into practice. Our more exciting program is developing this low-cost point-of-care ultrasound with University of Oxford, where simple sweeps of the probe of the ultrasound can be converted using AI into a traffic light decision system, which means one would not need ultrasound machines, but just portable ultrasound probes with no display, and be able to triage this information in resource limited settings. Again, this will move forward into the next phase of validation in a couple of years. And then we've come to a more complex system of actually looking at dynamic personalized risk prediction models, where what we've seen in the beginning as baseline predictors, using simple epidemiological methods, developed a prediction model. This is called a feed-forward method that we've been using. Then added some of the ultrasound parameters using complex machine learning, neural network techniques, and identified early predictors in the first trimester. And as we move forward, use some more of the anthropometry data that we've got from ultrasound to develop some more well-refined models for predicting preterm birth. But as you can see again here, that while there's a big jump from the baseline predictors to the early predictors in the ROC, as we move forward, it's plateauing off. And that is again where an interdisciplinary nature of moving forward comes in, because we need to put in more mixed technology there, more image technology there to improve this prediction model. And realizing our limitations, that this was something that our group may not be able to do on its own. Again, we were pushed by Dr. Bhan through the KI grants that BIRAC was promoting to partner with the data science team at IIT Madras to understand how this data could be analyzed to take it for understanding better prediction. Because we realized that for prediction preterm birth, you have to phenotype it well. And for phenotyping it, you need to understand the dating, the actual gestational age well. And that is where, partnering with them, now using our own anthropometry data, we've been able to develop models from our ultrasound anthropometry, which are working better than the global models available like Hadlock and Intergrowth for detecting gestational age in second trimester and even in third trimester. Again, this is initial evidence and needs to be validated in further independent cohorts. So here we had simple epidemiology, where we realized that we need more complex and more advanced methods to understand the prediction models. We are working on the omics technology to understand more predictive biomarkers, which not only which are used as biomarkers, but also go into a dynamic prediction model. We have image biomarkers, which again will give us tools for individual clinical decision-making tools, but also go into a dynamic prediction model. And all this would then help us to develop an algorithm, finally, which can go in as a public health setting for predicting preterm birth. Now, the steering committee that we had kept asking us that these are long-term goals. What is it that this cohort would give us as a risk factor, would give us for health policy interventions and integrate that can be integrated into social schemes? And that's when we started to look at some of the earlier deliverables that we can achieve from this cohort. We have seen, and as Neeta said, because we are studying in that, like they are doing in their cohort, we're following up our pregnant women so closely. We are being able to monitor their gestational weight gain, look at the fetal growth, and we have some very interesting initial data on how pregnant women are gaining weight during pregnancy and how the fetus is growing. And as we can see, 50% of pregnant women do not gain recommended weight. It's associated with fetal growth restriction. 1/4 of our babies are born growth restricted. We've been able to identify, and then is nutrition a problem? Because we have the opportunity of being able to study their dietary patterns and look at these dietary patterns changing across trimesters. So we've identified dietary clusters, and we are trying to evaluate their associations with micronutrient status, the protein quality, metabolic diseases, and fetal growth, so that in a couple of years, we are able to define comprehensively dietary advice, which is evidence-driven to optimize outcomes. Again, another important program which can go into public health is to understand how environment is affecting the baby, the fetal growth and early gestation. And as we said, we found that biomass fuel increases risk of preterm birth by 35%. We have now been able to, using low-cost sensors in collaboration with IIT Chennai, done a sensor-based temporal-spatial monitoring for air pollution in Gurgaon district, where our cohort is placed. And this is moving forward to do individual exposure of women during pregnancy to identify critical thresholds of exposure and windows and dose response relationship of air pollution in pregnancy, which would help us to guide interventions. And we're trying to study these with fetal biometry, looking at placental and urine metabolic changes to be able to understand mechanistic studies for intervention. So in the last 5 minutes, this is where the program is. It's still evolving, and there are a lot of unanswered questions that will be answered as we move forward. But during this, we've been able to develop and augment the research systems by setting up a state-of-the-art biorepository, which Dr. Swaroop mentioned right in the beginning of this session, which is now placed at the NCR BioCluster, coordinated by THSTI, which has over many hundred samples of the mothers and the neonates, and also ultrasound images from this study. So adding, and the other thing that we were asked by the steering committee is to use this platform to facilitate new hypothesis-driven studies. And this has led us to, as we move forward, to develop a very innovative program around the placenta. Placenta, because as all of us know, that it's an area which is like a black box of gestational events. You get it at the end of delivery, and so you really don't know what has happened to the placenta during the pregnancy, and it's actually the cornerstone of pregnancy. So that is a big biological program, along with the clinicians, that has started based on this platform that we've set up. Another one that we've been able to collaborate with our global collaborators on harmonizing our work with other cohorts that are going on in the country. So this is, just to summarize, that this is a huge, huge initiative, a program that is trying to answer a lot of questions where there's very little clarity. And this has only been possible because many teams have got together, many people are committed and dedicated to finding these answers, and with a strong support from the Department of Biotechnology at all times. And the two of our nodal officers who've helped us take this forward would not have been possible without them. And before I finish, I think all this has been possible because we had Dr. Bhan with us right through. And some of the messages that I would like to leave, some of the messages I'd like to leave for our younger investigators here, that your path is your responsibility. But when you find a mentor who enriches your life professionally and personally, the path can be transformative. And if they've opened the doors for you, make sure you walk through them. And this is what we learned from our mentor over these decades, 3 decades that we were with him. How do we do meaningful work? It's only getting out of your comfort zone, high risk, high award areas, accept big challenges, and like Neeta said, believe that we can't read the path alone, need to build bridges with different disciplines. There are a lot of areas in the platform that we've set up that we can't do alone. Even today, we are struggling to find answers. We get beaten at every steering committee meeting, at every review committee, and yet we move on because we believe that together we can actually find answers that we've started, because we've been taught that you finish the story that you have started, and we'll find those answers. Dr. Bhan was our biggest critique, but we all thrived on it. We thrived on that communication, on that, which was a constant learning, whether it was individual meetings, walks, drives, countryside, marketplace, informal get-togethers, seminars, work travel, formal meets, everywhere. We just absorbed and communicated and learned. And it was never as if it's going to stop. That learning continued constantly. And just for all the young investigators, the lessons that we learned: be an outlier, always be anxious about your success, that complacency kills. Don't worry about designations, only your work matters. So while Sir actually did what he did as a true mentor, but it's now for us to actually walk that path and finish what we've started. And I sincerely hope we can do it. Thank you.
Thank you very much, Ranjani. That was absolutely a stunning talk. As a matter of fact, Dr. Bhan welcomes me to my office every day. I have this particular picture stuck to my door. I remember him every day. We have, Dr. Bhan was very fond of young people and would train them and would set up collaborations for them, training programs for them. I'm not going to elaborate on that. I know some of it. He has a large number of people, young people, who for whom he has made these kinds of avenues possible. He has himself mentored many of them. And today we have two of the younger mentees of Dr. Bhan, one of whom has just completed his PhD. I would invite now Bireshwar Sinha, followed by him Ranadeep Chaudhary, to tell us about what they have learned from Dr. Bhan, but it's not going to be a personal recapitulation of their personal experiences with Dr. Bhan, more like academic experiences, and through these academic experiences we learn about the personality, about what Dr. Bhan stood for. Biresh.
Hi, sir. Can you hear me?
Yes.
Okay. I need to.
Yeah, share your screen.
Yeah.
Yes. Yes, fantastic.
Okay, thank you, Parthoda, for this opportunity to speak today about Sir. And as Sir would say, it was after passing MD and after doing the medical colleges, we used to call everybody, even a 1 year senior, as Sir. And Sir told us, you may call me Sir, but do not call everyone who is coming and talking to you a 1 year senior as Sir. So whenever I refer as Sir, I always mean Dr. Bhan. So he was an inspiration for us. And he was not only a great scientist, a visionary, but above all, to me, he is the best and loving mentor and guide one can ever have. So today I will try to divide my talk in two aspects. One is my personal acquaintance and experiences while working with Sir, and then I will try to summarize the key lessons that I have learned from him during the last 5 years. I have been working with Sir from 2014, and I will start from the beginning. So after my passing out, I got a call from one of the recruiting agencies, and there I first met Sir during my interview, and I heard from different places that at that time I didn't know about Sir, and all I knew that he was a very big personality and he was a huge person. But the first time when I met him, I found a cool guy coming inside and talking to me like a friend, and I didn't understand whether he asked me questions or how did my interview go. But I got selected, and then that started my tenure or my education with him. And so we were the basement gang. We. Sir used to call us folks, the young folks, and he used to come whistling down the way every time, and we knew, okay, Sir was coming, and we were so excited for the day because first he used to come down, he used to charge us with a morning high five. He used to come and greet each and every one of us by shaking his hands and giving us a high five. That kind of boosted our energy. And after that, we had this enriching discussions every day morning over tea and coffee. We used to talk about new ideas. And there was also a treadmill which was placed beside our table, where Sir used to do the treadmill exercise, and he used to give us with new challenges and ideas every time, and ask new questions. And he sometimes, when he used to come and sit on our table, we used to see notes written down by him, his handwritten notes about new questions or on a question that we were discussing about with Sir. For example, this was one concept note that we were trying to develop on azithromycin in third trimester and how it can affect birth outcomes, low birth weight, preterms. So he was giving his ideas, and he has put up some note on which obviously I needed to work on and take it forward and take feedback to him. He also kind of marked out articles, and he used to write names on these articles. One is for Biresh or one is for Ranadeep. So he used to segregate articles based on the domain in which each of us were trying to develop. So that was the kind of training and the kind of love he had for us always. And he had a way of explaining complex problems in a very simple way. And he used to give us these articles from Nature and Science, which sometimes was very difficult to understand because many of them were about newer techniques, methods. And whenever we had any questions, we used to go to him, and he had a unique way of explaining them so that we could understand. And previously, we used to have this pack meetings, I remember, and there was a pre-pack dinner. So Sir told, there is no free dinner, right? If you are getting all the people together, why not ask them a few questions and get feedback from them? He used to call them cute science meetings and workshops where we would kind of make these presentations, and it was really a very good experience on making these presentations with him. Because he told us about how to make the presentations in a simplistic way, but in a way where everybody can understand the issue, but the slides should not be too much crowded, and so he had a very simple way of presenting the slides. So that was the learning from here, and also the science meetings were very robustly planned during his time. So we used to have training sessions, practice sessions before doing the actual workshops where he used to criticize our presentations fiercely so that we were prepared for the same. Was the soul of the scientific meetings, and when we used to go to the science meetings with him, we were, yeah, sometimes we feel that we were kind of relaxed because we thought that, hey, Dr. Bhan is there, he will answer most of the questions. But I understand in the process of his training, he actually would train us so that we were well equipped to answer that. We understood that actually when we were standing there, we could actually answer the questions ourselves because we were kind of prepared with that. And he would always like us to question the questions. So he always told us that different ways to ask the right question. First, to structure the question in a format, in a PICO format, and then he would tell to send these questions first to the peers and to the best in the field and take feedback on the questions, on the novelty of the questions, the relevance of the questions, and the feasibility. He used to tell you need to send. He used to send us, tell us to send the email directly to people who were the best in the field and whom we were not acquainted with. For example, I remember I was writing something on the list model on diarrhea, and he said that you send your question to Bob Black. I have never known Bob Black, but he told you directly introduce yourself and you send the question. And interestingly, I didn't have the confidence, obviously, to send the question to Bob Black because I don't know him. He is at the John Hopkins and he's a big person. But what I understood is that if your question has some value and if the person understands that this person has thought about the question and has some depth in it, we get a response from the other side. So that was the learning experience that I had. And he was always available for us. He was, no matter how busy he is, whoever is coming or whoever is sitting in his room, if we had a question, he was always there to answer us. And and sometimes when he was busy and he was on the way to the airport, we used to sit inside the car and get time from him, ask our questions. So in the car on the way, he was always available. And he used to kind of call me his writing partner because I used to write a lot of proposals with him, and sometimes obviously during the initial days, I was kind of, I became sad when these proposals were rejected. And he kind of taught me that you should move forward and you should learn why your proposal was rejected, and you should move ahead, find another agency and then revise your proposal and then submit it again. And so we continued, and this was a fun exercise that yes, I think in the last 1.5 years, that part, because of various reasons, is kind of slowed down. But I think I need to gear up on that and start writing proposals again. And this was not only in the office. We had fun time in the evenings, in the cafes, at his home, and he was always accessible. We used to go together and discuss with him these new questions. And these were beautiful opportunities. And to me, also during the travel time, while we used to travel internationally in Bergen or in Geneva, so Dr. Bhan told us that when you need to travel, you see in the flight you have around 6 to 8 hours of time. You need to take at least 3-4 articles that you can cover up during your travel time. So that he told that is the best time where you can read undisturbed, because there is no one sitting around you whom you know. You can utilize that time and read these articles very peacefully, and you can absorb them. Apart from that, he was a fun person to travel with. You know, this I remember is a picture from Bergen, and we had a very good table tennis match with Sir during this time. It is in 2018. He was so Yeah, and he was playing really well, and it was a lovely match that I remember playing with Sir over here. Uh, he was also like a father and a friend, and this was 2 years back, and during November. Uh, and this is a time when most of the proposals were being submitted or had deadlines, or many meetings were going on. But I remember he told Madhu, his secretary then, that Madhu, you must remove or cancel whatever meetings is there on that day. It is Biresh's special day, and how can I not be there? He was like a father to me and also like a friend. And then the whole family of SAS was there to support. It was a lovely moment. It is like being together, the sense of togetherness. Uh, so he was the light of our lives and always full of energy. And I must tell you, he was kind of the youngest person of the basement, and we did, and his metabolic age, you will be surprised that his metabolic age in 2019 was 35, and almost all of ours metabolic age were kind of more than him. So he was kind of the youngest in the basement. So the 10 lessons, if I want to summarize the 10 lessons that I have learned or I try to follow from him, and so the first lesson that he taught is never settle for anything but the best. He always told us that you have to be above the 90%. You have to be an outlier, as Dr. Shinjini already said. He said that you have to be a fighter and a leader. And the second lesson is that deep reading. You have to. So based on his feedback, we created every one of us. I think we have created now a journal base, which we screen periodically. We screen this journal and screen out the relevant literature, and so that we can stay updated with the new. The third that he taught us is ask as many questions as possible and use a format that is the PICO format for asking the detailed questions. And you have to assess the questions on the terms of relevance, its feasibility and novelty. And he told to pursue the ones which are only greater than 8 out of 10. The fourth lesson that I remember is to seek feedback from the peers and from the best of the field. So he said not to be afraid, and if you, from the literature, if you see somebody is working in that field, you must directly reach out to that person for feedback. And he wanted us always to learn from the best. And he told us to look for the best courses in the world, and you need to learn from the best and do it yourselves, hands-on. And he told there is no compromise on the scientific methods. And whenever we were doing our PhD studies or other scientific studies that we were planning, when we had these review meetings, he was our biggest critic. He used to challenge us, question us, and he used to push us to make sure that the methods, the laboratory methods and the statistical methods are world class. And he always pushed to stick to the best practices. And one thing that I remember, that while writing proposals and also otherwise, he used to say that think of the science and administrative issues separately. We were sometimes stuck between making a budget and all those things. So that was his advice to us. And then he was very specific about that we need to choose a career path early, a scientific domain of expertise, while continuing to read broadly about the other aspects. So for me, I kind of, though in the initial days I used to work more broadly, currently I chose my domain around the infection nutrition, mainly focusing on infectious diseases. So he wanted us all to choose a specific domain so that we can read more deeply within that domain, but also need to have some broad reading side by side. The eighth lesson that he told us is to teach what you read. So he told that we need to speak out. So we need to teach our juniors or kind of convene workshops where we take the classes so that we can express ourselves clearly. And he thinks that is the best way to learn. He told to write avidly, revise and revise the manuscripts until you are satisfied with yourself. So that is a practice which I try to follow as much as possible. The ninth lesson is don't stop and move ahead. He taught us to learn from failures, understand the reason of the rejection, and improve on that. And the most important lesson that I think he taught is about working together as a team. He told, and I would quote, that alone you can go that much far, but together you can accomplish anything. Above all, he always told us, do not forget to smile, because that is how you can collaborate and work with everyone together. So this is what I have learned so far from him, and I'm very happy to be a part of this great team. All of us at SAS, and not only SAS, all the collaborators at other institutes, at KEM, at CMC, at NIBMJ, THSTI. So I'm lucky to be part of this great family, and thank you, Partha, for this opportunity. I'll stop here.
Thank you, Biresh. Thank you very much. The 10 lessons were also lessons for us, for many of us, even at this age. So thank you very much for enunciating those. Our next young folk is Ranadeep Chaudhary. Ranadeep also did his PhD with Dr. Bhan, and he continues to do work that Dr. Bhan had initiated, and he's, of course, taking it forward in more ways than one. He's just been awarded with a Wellcome Trust DBT India Alliance grant. Ranadeep.
Yeah, thank you, sir. I don't have any slides. So I was just thinking what to say today. And then suddenly one thought came to my mind that why India doesn't produce so many biomedical or world-class biomedical researchers. Then I thought that maybe we don't have the culture in most of the medical schools. And another could be that most of us don't get that opportunity to get a mentor like Dr. Bhan. So today, in next 5 minutes, I will not talk too much about Dr. Bhan's contribution to science or betterment of Indian science, because I think many of you know better than me. But what I will talk about, that how Dr. Bhan can motivate or direct or show path to a recent pass out medical graduate or postgraduate who doesn't know exactly what to do after completion of MD. But one thing he knows, he knew very surely, that he doesn't want to do clinical practice, and a person like me. And when I see my career path over last 7 years and having interacted with many people, what I've understood that this is not only me. There are many people in India, or many medical graduates or postgraduates in India. They really don't know what to do after MD, but they don't always get Dr. Bhan. Then why, how Dr. Bhan have shaped my career and could have, and also may have shaped many others' career? Why? How? Because Dr. Bhan, in true sense, was a great mentor. And a mentor is different from a coach. A mentor is different from a guide, because mentor always tried to synthesize what one wants and shape his or her career in that way. So one sense, a mentor extract exactly what the mentee wants to do, and the way Dr. Bhan shaped my career in that way. What he inculcated me, and I think he inculcated that in many of us or many of the young folks, that the passion of reading, and passion of synthesizing, and connection, and then synthesizing connection and develop new research question, and develop new research questions of two things: of national importance, and of to understand one biology, to one understand the domain of that biology. So this issue is not a single day issue or a 2 day issue. This issue became developed, or rather developed interacting days, interacting with hours, days and nights. And thereafter these issues come. So all these things Dr. Bhan had inculcated me, and I must say that I along with Biresh and two other of my colleagues who are, I don't know whether today they are here or not, Ravi and Tarun, we four are, I think, were the last were our mentors for last 6 or 7 years, and we are the luckiest one among the lot whom Dr. Bhan mentored and showed the path of our future. That and another one important issue I want to tell all of you today on this day, that it's not a easy thing, or this is not a very single step to do. People can born with talent, but talent without hard work, talent without reading doesn't mean anything. And all these issue Dr. Bhan inculcated into all of us. In this lockdown period, in this pandemic time, this year I achieved two professional milestone in my career. I completed my PhD from University of Bergen, and then I also got awarded one of the most prestigious fellowship of Welcome DBT India Intermediate Fellowship. Dr. Bhan was not that physically when I was awarded these two, but maybe his blessing is with me, and I'll stop here. Thank you. And thank you, Parthoda, for giving me opportunity to talk about Dr. Bhan and his contribution for the young folks. Thank you.
Thank you very much. I think we've come to the last 2 or 3 minutes of today's program, an exposition of science that was initiated by Dr. Bhan and has been taken forward by many of his students and close colleagues. The take-home message from here is that we need to follow some of the principles and some of the culture that Dr. Bhan actually enunciated and that some of us have imbibed. I really think that great men who have departed leave behind footprints, very strong footprints on the sands of time and sands of institutions such as in IBM, GT, HSTI, DBT, etc. So I really want to take this opportunity to thank Dr. Renu Swarup, who is, I don't think has been able to join, Neeta, Gagandeep, Shinjani, Biresh and Ranadeep, and also our director, Professor Soumitra Das, for introducing and welcoming all of you. I will leave the podium now and have Dr. Soumitra Das have the last word. Soumitra.
All right, so I'm sure all of us actually paid the real tribute, and I'm certain that Dr. Bhan is actually watching from the top, as you have rightly mentioned, this legendary scientist and the Maharaja of the translational research. So we have in our small way given the tribute, and we all need his blessings to move forward. So he has left his legacy, and we have this now responsibility to carry on this legacy. And I'm sure the people whom he trained and people who are blessed with his association are actually efficient enough to carry out his legacy and will actually really contribute to the world of science. Thank you once again for taking trouble to actually participate. And in the online version, this is something, again, Dr. Bhan's blessings. It is actually flawless event, because many a times we have some technical glitches and other things, but today, with his blessings, we didn't have any problem, and we have really enjoyed the scientific sessions and as well as the down the memory lane from his associates. So with, again, my personal tribute and on behalf of the National Institute of Biomedical Genomics and all of you, we pay one more time the homage to this departed soul, and I'm sure we'll do a good job and we'll take his blessings and move forward. Thank you all. Thank you all.
Thank you very much. Let me also take this opportunity to thank, because you have mentioned that we have done a technical job which had no glitches at all. Let me thank Mr. Bikram Roy, our IT manager, and his team for this flawless event. Thank you very much, Bikram.
Thank you.
Thank you.
Thank you. Thank you very much.
Thanks, Siri. Thanks, Rita. Thank you. Thank you, Parthuda.